Hi everyone, let's move on to a new family here in Chapter 11. This is the Enterobacteriaceae, or the Enterics. The Enterics are organisms that obtain energy by anaerobic respiration, also known as fermentation. They use nitrates, sulfates, and carbohydrates for energy. So let's go ahead and talk about the Enterics. Just kind of a generalization, this is usually the flora of your guts and butts. So enterics are often referred to as coliforms, and coliform is just a general term for enteric. A large number of organisms live in this family, and they're actually broken down into tribes, and we're going to talk more about that later. All of them ferment glucose, and some produce gas, but others do not. You can actually see that gas is produced in a slant known as a triple sugar iron agar. Klebsiella pneumoniae is the most vigorous gas producer. All of these organisms will reduce nitrates to nitrites, so they're going to reduce NO3 to NO2. Only Plesiomonas will produce cytochrome oxidase. So we won't talk about Plesiomonas too much in this material, but I just want you to know that all the rest of the enterics are cytochrome oxidase negative. And you probably remember the oxidase test from our unit on Neisseria and Moraxellas. All of the enterics are motile except for Klebsiella, Shigella, and Yersinia, and these are motile at body temperature, but this can be tough to demonstrate. So usually we set up an organism's motility at body temperature and at room temperature to determine where they are motile at. And none of the enterics are fastidious. They all grow just fine aerobically, even though they are facultative anaerobes. These enterics are the first gram-negative rods that we're going to talk about. Our Neisseria and Moraxellas were gram-negative diplococci, so this is the first actual rod that we've talked about. They're gram-negative rods. They're usually short and chubby looking on the gram stain, and they are the most frequently encountered bacterial isolates in clinical specimens. They are found in the intestinal tract of mammals and humans. They also can be found in the soil, water, and plants, so they actually are found in the environment that we live in. Humans can be infected by enteric contaminated food or water, and when the intestinal microbes migrate away from the gastrointestinal tract. So these pathogens are virulent because they produce toxins, they're very antibiotic resistant, or they can be very antibiotic resistant, and they tend to cling to our tissues and colonize us. I want to describe the tribes, even though you do not need to memorize what tribes each organism is in. But tribes are a way to place biochemically similar organisms together. There are so many species within the Enterobacteriaceae family that we have to put them into tribes. So as you can see here, tribe 1 is our Escherichiae, tribe 2 is our Edwardselliae, tribe 3 is Salmonelliae, tribe 4 is Citrobacteriae, tribe 5 is Klebsiella, tribe 6 is Proteae, and tribe 7 is Yersinia. So I just want to talk a little bit about which organisms are in which tribe in the next slide. So tribes include the organisms, these tribes include organisms in each tribe. Escherichia and Shigella are metabolically similar to one another, and so they are in tribe 1. Edwardsellia is in tribe 2, Salmonella is in tribe 3, Citrobacter is in tribe 4, Klebsiella, Enterobacter, Hafnia, and Serratia are in tribe 5. Tribe 6 would include Proteus, Morganella, and Providencia, and then tribe 7 includes the organism Yersinia. Now, bacterial identification is completed by evaluation of how bacteria react with organic compounds. So, for example, if we're looking at oxidation reduction, Pseudomonas is an oxidizer, and an enteric is a fermenter. Some tests that we use to identify these organisms is also carbohydrate utilization and formation of acids. For example, the OF glucose test, are they fermenters or oxidizers? We also can use gas production as a way to identify organisms. We can use products of fermentation, or what types of acids are produced. We can look at carbon source tests and carbohydrate utilization and formation of non-acid metabolites, like in the Voges-Proskauer test. We can look at specific enzyme tests like coagulase, deaminase, etc. And then finally we can look at inhibiting agents like sodium chloride, bile salts, and potassium cyanide. But these are all ways or tests that we can use to identify our organisms in each one of these tribes. There's a number of different culture medias that we use to isolate and identify our enterics. We have what's known as mildly selective media, moderately selective media, and highly selective media. Some of the mildly selective media that we use is MacConkey agar, EMB, and MAC with sorbitol. And some moderately selective and differential media would be Salmonella and Shigella agar, Hektoen Enteric agar, and XLD agar. One type of media that is highly selective that isn't on this slide is called brilliant green agar. Make sure that you look at Appendix A in your textbook because it gives a nice description of all the different medias that we talk about in lab. There are also a couple enrichment broths that we use in the isolation of enterics, especially if you're looking for Salmonella or Shigella. If we get a stool culture in the laboratory and we are looking for enteric pathogens like Salmonella or Shigella, we might set up a selenite broth or a gram-negative broth. A selenite broth is set up in addition to your plate media in stool cultures, and it's recommended for the isolation of Salmonella from feces, urine, or sewage. Gram-negative broth is used for Salmonella, but it's actually a better media to find Shigella, and it inhibits their gram-positive bacteria. It may inhibit Proteus and other enterics and increase the isolation of Shigella. When trying to identify our enterics, we want to look at the fermentation of lactose, sucrose, and sorbitol because the fermentation of glucose is not helpful because all enterics ferment glucose. The macroscopic appearance of enterics on blood agar is of little value because they all appear to be dark, damp, gray, and gamma hemolytic, except for maybe E. coli, which could be a beta hemolytic organism. Some of the medias we use to isolate our enterics would be selective medias like MacConkey, EMB, Salmonella Shigella agar, Hektoenteric agar, XLD, and finally MAC with sorbitol. These medias contain lactose, sucrose, and sorbitol, and xylose. They all help to differentiate enterics by carbohydrate fermentation. Don't forget, all enterics ferment glucose. Here I have another slide that you do not need to memorize, but it's going to be helpful for the recognition of organisms. So I would definitely use this chart when you're dealing with organisms in lab. We have some organisms that are lactose positive and other organisms that are lactose negative. Carbohydrate fermentation should be viewed on slants and plates at less than 24 hours, and we always want to look at our isolated colonies. Some organisms that are lactose positive would be E. coli and E. coli 0157H7, Enterobacter aerogenes, and actually the new name for that is Klebsiella aerogenes. I need to change this slide. Enterobacter cloacae and Enterobacter sakizaki are all lactose positive. Klebsiella pneumoniae and Klebsiella oxytoca, and finally Citrobacter frundii. These are all organisms that should be pink on our MacConkey agar, and they would be dark opaque purple on our EMB agar because they are lactose positive. Our lactose negative organisms would be Proteus mirabilis and Proteus vulgaris, Morganella, Salmonella, Shigella, Dysenteriae, Boydii, and Flexneri, and finally Yersinia. These are all organisms that are going to be colorless on MacConkey agar, or they are going to be clear and colorless on EMB agar. Now, there are some organisms that slowly ferment lactose, and this will take place after 24 hours. And some of these organisms would be Serratia and Hafnia. They're actually considered what we call ONPG positive. They're slow lactose fermenters, and that's determined by the ONPG test. And we're going to discuss this later, but don't forget, ONPG will always be positive for your rapid lactose fermenters. And then here on this slide, again, you don't need to memorize this, but it's helpful to know which organisms are sucrose positive and which organisms are sucrose negative. Okay, this can be determined if sucrose is in the media, like Hektoenteric and XLD media both have sucrose in it. If you have organisms that are sucrose positive in the HE media, the colonies turn salmon or orange. If you have organisms that are sucrose positive in XLD, the colonies turn yellow. Now, MacConkey agar, Salmonella Shigella agar, and EMB agar don't have sucrose in them, so we cannot evaluate sucrose fermentation on those medias. But some of the organisms that are sucrose positive would be E. coli O157H7, Citrobacter fruendii, our Enterobacters, Klebsiellas, and Proteus vulgaris. Some of our organisms that are sucrose negative would be Shigella, Morganella, Providencia, Serratia, and Salmonella. So let's take a look at our sorbitol fermenters. So on this slide, I've outlined which organisms are sorbitol fermenters and which ones are not sorbitol fermenters. And really the only one that you need to know on here is that E. coli O157H7 is sorbitol negative. That's pretty much the only reason why we use MacConkey with sorbitol plates. We are looking for strains of E. coli that are possible O157H7 strains because that can lead to a disease known as hemolytic uremic syndrome. MacConkey agar is designed to differentiate those enterics based on their ability to ferment lactose. Our organisms that grow on MacConkey agar that are lactose positive will turn pink, and organisms that are lactose negative will be colorless. There are bile salts and crystal violet that actually inhibit the growth of gram-positive organisms on this media. The pH indicator in this media is called neutral red, and that's what allows our organisms to be pink if they are lactose fermenters. And then here on this plate, we can see we have a kind of an array of lactose positive and lactose negative organisms. So the pink colonies, if you look out there on the isolated colonies on the bottom left, are the lactose positive colonies, and the lactose negative colonies are the ones that are colorless. And there's two kinds of organisms on there that are colorless colonies, so they are lactose negative. Usually we're concerned most with Salmonella and Shigella that are lactose negative. Other medias that we use in the microbiology lab are EMB, MAC sorbitol, and SS. EMB is eosin methylene blue, and here we have eosin and methylene blue that are the pH indicators in the media. Vigorous fermenters of lactose may be metallic green. One organism that can be metallic green on EMB is E. coli. Others that are lactose positive are going to be kind of this opaque purple color. MAC-S is a media that we use to see or evaluate sorbitol fermentation. If you have an organism that is sorbitol positive, the colony colors are going to be pink, and if you have organisms that are sorbitol negative, they're going to be colorless. The one that I think about most is E. coli O157H7, and it is sorbitol negative, so it would be a colorless colony on MAC with sorbitol. And then finally we have Salmonella and Shigella agar. This inhibits gram-positive growth because it has high bile salt content in the media. It also contains neutral red, lactose and sodium thiosulfate, and ferric citrate for the detection of H2S. So in this media, if you have any colonies that are pink, they are lactose fermenters. If you have colonies that are colorless, they are non-lactose fermenters. And we can also determine if our organism produces hydrogen sulfide on this media. If our organism produces hydrogen sulfide on SS agar, then they will have a black center. So let me show you some pictures of the media in the next slide. So in this image we have EMB agar, and you can see that we have a metallic green organism growing on that media. That is most likely to be E. coli because E. coli is a vigorous fermenter of lactose. On MacConkey sorbitol media, any organisms that are sorbitol fermenters are going to be pink, and our sorbitol negative organisms will be colorless. On Salmonella Shigella, we have lactose in that media, and our lactose fermenters are going to be pink, and our lactose negative colonies are going to be colorless. Some of the moderately selective media that we use in the microbiology lab are Hektoen Enteric agar and XLD. Hektoen Enteric agar has bile salts and bromthiamyl blue in it, and also acid fusion. This inhibits the growth of gram-positive organisms. So on HE, you're only going to get your enterics to grow, and we can determine if those enterics are lactose or sucrose fermenters. If they're lactose or sucrose fermenters on HE, they're going to be salmon or orange colored. Okay? On HE, if they are lactose non-fermenters, they're going to be a clear green color. XLD is a moderately selective media for the growth and identification of enterics also. It contains sodium deoxycholate, which inhibits gram-positive growth. It also contains the sugars lactose, sucrose, and xylose. All enterics ferment xylose except for Shigella. So lysine in the media prevents Salmonella from rapidly fermenting xylose and appearing like a Shigella. Okay, so only very small quantities of xylose are noted in XLD media, but if you have an organism that is positive for lactose or sucrose or xylose fermentation, the organisms are going to be yellow, and if they are lactose, sucrose, or xylose negative, the colonies will be clear red. So using these moderately selective media, in addition to your mildly selective media, using those plates in combination with other plates, helps us identify the organism and which sugar has been fermented. We can actually deduce which sugar has been fermented by using a combination of plates. So whenever we are setting up a stool culture, we always use mildly selective media and also a moderately selective media. Because HE and XLD are also moderately selective medias, we also can determine if our organism is a hydrogen sulfide producer. If our organisms are hydrogen sulfide producers, those colonies on HE and XLD will be black in the center. So for example, if I have an organism that is a lactose fermenter and also an H2S producer, the colonies will be yellow with a black dot. If you have organisms that are lactose or sucrose positive colonies on XLD, those organisms will be yellow. If they are hydrogen sulfide producers, you will get a black dot in the center of those colonies as well. So let's take a look at this on the next slide. Some medias are very sensitive to H2S production and others are not as sensitive. As you can see here on the XLD plate, the Salmonella is lactose and sucrose negative, so the colonies are clear red, but there's also a black dot in those isolated colonies too, indicating that they're H2S positive. Okay? In the image on the right, this is a sulfide indole motility agar, and we can see the black line in the center. That indicates that hydrogen sulfide production has taken place as well. We can actually see the production of hydrogen sulfide because of the components sodium thiosulfate and ferrous sulfate that are the H2S indicators. Sodium thiosulfate is a gas that can be visualized using ferrous sulfate. Not only can we evaluate fermentation of different carbohydrates on plate medias, we can also determine the sugars that have been fermented on a tube media known as triple sugar iron agar. Triple sugar iron agar has 3 carbohydrates in the media, glucose, lactose, and sucrose. And we can determine if an organism produces hydrogen sulfide on TSI as well. The source of the hydrogen sulfide in the tube is ferrous sulfate and sodium thiosulfate, phenyl red indicators, and peptones. So let me show you in the next slide what these TSI reactions will look like depending on which sugar is being fermented and what it'll look like if there's H2S production. So these are triple sugar iron agars, and they usually start out like the one on the right where they are red. That is also described as alkaline over alkaline, but the media starts out like that. If you have an organism that ferments glucose, you're going to see yellow at the bottom of the tube. Okay, so you can see in that first tube on the left where it's yellow throughout the tube, we call that acid over acid gas. So acids are being made because glucose has been fermented in the bottom of the tube, and then in the slant of the tube, lactose and or sucrose have been fermented. In the second tube, you can see that it's alkaline over acid or red over yellow. Because it's red over yellow, it means that lactose and sucrose have not been fermented, because you would observe that in the slant of the tube, but glucose has been fermented, and you can see that the bottom of the tube has turned yellow. In the third tube, we have an example of alkaline over acid plus H2S. We can assume because it's an enteric that it's actually glucose positive at the bottom or yellow at the bottom, even though we cannot see that taking place because it's been obliterated by the H2S blackening of the media. But it is actually alkaline over acid where no lactose or sucrose has been fermented, but glucose has been fermented and there's H2S production noted. And then finally in the last tube on the right, this would probably not be an enteric because all enterics ferment glucose. And you can see at the bottom of the tube on the right that there is no glucose fermentation or no yellowing at the bottom of the tube. There's also no yellowing in the slant of the tube. So this is considered a non-lactose, non-sucrose, and non-glucose fermenter. So it must be some organism like Pseudomonas aeruginosa. It cannot be an enteric. So here's the same image, and on this image I basically have outlined what sugars have been fermented and what sugars have not been. So again, at the base of your tube, anytime it is yellow at the base of your tube, we know that it's a glucose fermenter. If it is yellow in the slant of the tube, then we can say it's either a lactose or a sucrose fermenter. We don't know for sure unless we compare our TSIs to our plate medias, okay? But in that first tube, it is yellow in the slant, so it's a lactose or sucrose fermenter, and it's also yellow in the butt of the tube, which means it's a glucose fermenter. You can also see a space at the bottom of that first tube on the left, and that is a space because of gas production, okay? You can also see in the second tube that we have a alkaline over acid result. So there is no lactose or sucrose fermentation. You can see the slant is still red or alkaline, but the bottom of the tube and the butt of the tube of that second tube, it's yellow. So that is a glucose fermenter and only a glucose fermenter. In the third tube, it's alkaline over acid plus H2S. So we can say that it is not a lactose and not a sucrose fermenter because it is red in the slant. But even though we can't see the yellow at the bottom because of the H2S, if it's an enteric, it must be glucose positive. And then finally, in the last tube on the right, it's alkaline over alkaline. Because the slant is still red, there is no lactose or sucrose that has been fermented, but glucose has not been fermented either because it is also red at the butt of the tube. That's a lot to take in. I would definitely slow this slide down and write out kind of what I just explained. Now here we can use a combination of plates and tubes to determine what sugars have been fermented. So because MacConkey has lactose in it, you can see that these organisms are lactose fermenting because they are pink. If you look at the tube, which is the TSI on the right, you can tell that lactose has been fermented because the slant is yellow, okay? And don't forget, all enterics are glucose fermenters. So if I were to compare these, I could tell that my organism is a lactose fermenter. Now it may be a sucrose fermenter, but we can't tell because we don't have a plate that has sucrose in it. The ONPG test is a test that determines if an organism is a slow or delayed lactose fermenter. These organisms have beta-galactoside but lack beta-galactoside permease enzymes. You can read more about this in your textbook. But the ONPG test, that acronym stands for ortho-nitrophenyl BD-galactopyrroside. And rapid lactose fermenters must possess two enzymes, beta-galactoside permease and beta-galactosidase. Okay? Permease actually allows lactose to penetrate the cell walls. True slow lactose positive organisms lack this enzyme. Beta-galactosidase produces glucose and galactose. A yellow color is positive in the ONPG test, colorless is negative. So if you have an organism where the test turns yellow, it means that lactose has been fermented but slowly. So let's take a look on the next slide at all of the tests that we can use to identify our enterics down to the species level. So here's all the metabolic and amino acid utilization and miscellaneous tests we use to identify enterics. We can use the Methyl Red test, the Voges-Proskauer test, decarboxylase tests, deaminase tests, citrate tests, gelatin tests, indole production tests, malonate utilization tests, motility tests, urease activity, nitrate reduction, and then the growth in the presence of potassium cyanide. These are all the tests that we can use to identify our enterics down to the species level. So the first test I want to talk about is the Methyl Red test. And this determines the end products of glucose metabolism by using a dextrose broth. We use a broth known as MRVP broth because we do the Methyl Red test and the Voges-Proskauer test in the same broth. Glucose is metabolized using the mixed acid fermentation pathway. This test can actually take 18 to 72 hours for it to work. Bacteria are incubated in this dextrose broth or glucose broth, and stable acid end products are produced, resulting in a red color of the broth after the addition of the methyl red reagent. The principle is this: if glucose is metabolized by mixed acid fermentation, stable end products result in a pH of less than 4.4, so your methyl red reagent will turn that tube red. So let me show you a picture of this in the next slide. So the tube on the left is our MRVP tube that has been uninoculated, and that is just a dextrose broth. If it is growing, it'll get cloudy like that tube in the middle, and then if we add our methyl red reagent, if it turns bright red, it is positive. If it doesn't turn any color at all, it is negative. All enterics are methyl red positive except for Klebsiella and Enterobacter. Okay, Klebsiella and Enterobacter are VP positive, and you're going to see that most organisms that are methyl red positive are VP negative, and most organisms that are VP positive are methyl red negative. But that's not always the case. There's always those kind of stepchildren like Shigella and Serratia and Hafnia. Those are kind of the children that don't like to play nice. The Voges-Proskauer test is done in combination with the methyl red test and detects the presence of acetyl-methyl carbonyl as an end product of glucose metabolism. We take 40% KOH and alpha naphthol and add it to a dextrose solution. A red color after room temperature incubation is equal to a positive test. Most organisms, if they are MR positive, are VP negative and vice versa, but not always. Like I said in the last slide, Hafnia and Serratia can be odd and variable. Anytime we add our 40% KOH and alpha naphthol to our dextrose media, we want to allow that tube to sit for 10 to 20 minutes before we interpret the results. When a carboxyl group is removed from an amino acid by an organism, this is called a decarboxylase test. Okay? This is an anaerobic process viewed in the butt of the tube. And we use several amino acids in microbiology to determine if they can decarboxylate them. For example, there's what's known as the ornithine decarboxylase test, and we can determine that in an MIO tube, which is motility indole ornithine tube. Or we can do the lysine decarboxylase test that is determined in the lysine iron auger slant. But just understand that these are anaerobic processes that are viewed in the butt of the tube. So some bacteria possess enzymes capable of decarboxylating amino acids like ornithine and lysine, and this all happens in the butt of the tube. Okay? Tubes that we use are motility indole ornithine auger and lysine iron auger. So here's some images of lysine iron auger, and you can see the first one labeled C on the left is actually an uninoculated media, okay, where it's purple over purple. Okay, decarboxylation happens in the butt of the slant. So if it's purple at the bottom, that is a positive test. If it is negative, that's going to be yellow, which is a negative test, okay? And these media, the LIA media, can also determine if we have H2S production. So you can see in the LIA slant on the very right in column number 4 is purple over purple with black. So it'd be lysine decarboxylase positive because it's purple and H2S positive because it's black. Another amino acid utilization test that we can do is called the deaminase test. Here we're looking for the removal of an amine group from an amino acid. This is an aerobic process that is viewed in the slant of the tube. Amino acids can be metabolized by deaminases. These are enzymes that remove an amino acid or an amino group from an amino acid. This is an aerobic process that happens in the slant of the tube. Modifications to the deaminase test are phenylalanine deaminase and lysine deaminase. Deaminase tests determine if the organism has the enzyme present to deaminate phenylalanine to phenylpyruvic acid or lysine to cadaverine. So there an amine is removed from either phenylpyruvic acid or amino group is removed from lysine to form cadaverine. So here's an example of a phenylalanine deaminase test. We have a phenylalanine slant, okay? The phenylalanine deaminase test is the same thing basically as tryptophan deaminase. So sometimes you'll see this abbreviated PDA or TDA, depending on whether we're talking about phenylalanine deaminase or tryptophan deaminase, but essentially they are interchangeable reactions for microscan, API, and similar test assays. Both tests determine if we have an organism that has the enzyme that converts media products to alcohol and ammonia. So here in the phenylalanine deaminase test, we are looking at the slant of the tube because this is an aerobic process. So if our organism grows on the slant of the tube and turns green with the addition of 10% ferric chloride, then it is positive for phenylalanine deaminase. And you can see that in the image. We have E. coli on the left and Morganella morgani on the right. Our E. coli is negative. It does not turn green in the presence of 10% ferric chloride, but you can see that our Morganella morgani is dark green. It is a positive test for phenylalanine deaminase. In this picture, we can see that lysine has been deaminated here as well. Notice how the slant of the tube has turned wine color. If it's wine in the slant, it means it's a Proteus mirabilis or a Providencia stewarti. Those are the only two enterics that will actually deaminate lysine. But again, we're looking at the slant of the tube and we're looking for a wine color. The citrate utilization test determines the ability of an organism to use sodium citrate as a source of carbon for metabolism and growth. Carbon is absolutely essential in bacterial nutrition. Carbon, nitrogen, and an energy source is what bacteria need to survive. There is an organism known as Citrobacter, and that's going to be positive for the citrate utilization tests, but so are Enterobacter and Klebsiella. Organisms will grow on the citrate media, causing the pH to become very alkaline. If our tube starts out as hunter green and becomes kind of a royal blue, it indicates that there are alkaline byproducts and that it would be a positive test for citrate. When we do this test, we use very little organism to perform the test because all organisms will eventually leave dead carbon skeletons and we don't want false positives. So instead, we use a little bit of organism, we put it on the citrate slant. If the citrate slant turns blue at all, it is positive for citrate utilization as its sole source of carbon for energy. Bacteria who possess the gelatinase protein will quickly break down proteins like gelatin into amino acids. So the gelatin test determines the proteolytic effect of bacteria on gelatin. Due to the enzyme gelatinase, some bacteria can break gelatin down into a liquid substance. What we do is we inoculate the gelatin media with the organism and incubate it for 35 degrees at 24 hours or so, and then we refrigerate it and then we invert it. If it has become a liquid, it's going to slurp down to the bottom of the tube. Serratia and some low-frequency proteas can actually do this. Indole is a degradation product from the metabolism of the amino acid tryptophan. Bacteria possess the enzyme tryptophanase if they have a positive indole test. Indole production can be detected in the tryptophan media by observing for a red color after we add a reagent known as the Kovacs reagent. Kovacs reagent is also known as Ehrlich's reagent or 10% para-dimethyl aminobenzaldehyde. Do you have to spell that? No, but you're going to have to recognize that in a testing situation. Ehrlich's reagent is actually very similar to Kovacs, but it's a little bit more sensitive for non-fermenters as opposed to enterics. So here's an example of a negative broth right in the middle, or actually that's an uninoculated broth right in the middle. That is a broth that has tryptophan in it. And if you look at the tube on the left that's got the red ring around the top, that is actually a positive test for indole, where we've added our 10% para-dimethyl aminobenzaldehyde or our Kovacs reagent. And then if you look at the tube on the very right, this is a negative Kovacs or indole production test. The malonate test is actually a utilization and fermentation test. We're looking to see if our organism can use malonate as a source of carbon. Organisms that use malonate will grow in the media as a source of carbon, and the media will turn blue if it's positive for malonate utilization. The end product is sodium hydroxide. Ammonia salts are what turn the media blue. These organisms also use ammonia salts as their nitrogen source. So let's take a look at what malonate looks like in a positive test. So if you look at the broth on the left, how that broth is turning blue, that is a positive malonate test. And then the one on the right is negative, that is colored green, that is a negative malonate test. Most often our Klebsiellas are malonate positive, but our organisms can be positive for citrate and malonate both, or negative for malonate and citrate both. So most of our enterics are motile. Our Klebsiella and Yersinia and Shigella are not motile, but most of the other ones are. We can do a motility study to determine flagellar movement of our organism. Now, usually motility is viewed at both body temperature, which is 35 degrees Celsius, and room temperature. Okay? We use a semi-solid media like motility indole ornithine agar or sulfide indole motility agar, and what we do is we stab that media with our organism, and then we incubate it with our caps loose. Then what we do is we look for a hazy appearance around the stab. If you have a hazy appearance around the stab, it means that our organism is motile. And that could be in the room temperature the room temperature tube, or it could be in the body temperature tube. So let's take a look at an image of motility positive. So on the left, this media has been stabbed. I don't see any hazy appearance around that stab. So on the left, that organism is non-motile. But on the right, you can see a hazy appearance where it was stabbed right down the middle. And that indicates that our organism is motile. And like I said, most of our enterics are motile. In the urease test, we want to see if our organisms will break down urea. They can actually break down urea into end products that are ammonia, water, and CO2. So organisms that have the enzyme urease will hydrolyze urea, releasing ammonia and producing a pink color. So this media starts out as kind of a peach color, and it becomes pink if our organism has the enzyme urease. There are several different types of urea medias available. Some are slants known as Christensen's urea, and some are broths, and the broths are very sensitive to small amounts of ammonium being released. So here's a picture on the left of our urea broths, and you can see that one is positive, very pink, and the other one is negative. And then on the right, we have our Christensen's urea slants. Any pink at all is considered positive, and they start out kind of peach colored. All enterics are nitrate reduction positive. If you recall from several units ago, our Staphylococcus are nitrate reduction positive, but our Streptococcus are nitrate reduction negative. All enterics are going to reduce nitrates to nitrites or nitrogen gas. In a nitrate broth, we can incubate our organism and then add our two reagents, sulfonylic acid and alpha-naphthylamine. If the tube turns pink, then the organism has reduced nitrates, and that usually happens within about 5 minutes. If the tube does not turn pink, or red anyway, then we're going to add zinc. If the zinc turns the solution pink, it means that nitrates are still present and have not been reduced. If the zinc does not turn it red, that is still a positive test, only this time nitrates have been reduced to nitrogen gas. In this image, we can see two positive nitrate reduction tests. On the left, we have what our enterics would do. We take a nitrate broth and we add equal amounts of sulfonylic acid and alpha-naphthylamine. Usually in a big tube, I add 8 drops and 8 drops, and then I allow it to sit for 5 minutes. If it turns red or dark pink, it is a positive nitrate to nitrite reduction test. If you add zinc, if it's negative and you add the zinc and the zinc does not turn it pink, it is still a positive test for nitrate reduction. It's only reduced nitrates to nitrogen gas, and that's what you can see in the image on the right, in the tube on the right. Finally, the last test that we can do to identify our enterics is the potassium cyanide test. It is not done routinely anymore because of the health risks to laboratory professionals. But this test determines if an organism will grow in the presence of potassium cyanide. Klebsiella and some of our Enterobacters will actually grow in the presence of potassium cyanide. Okay? Now, at the end of this unit, you should have in your packet examples of tests for speciation and how organisms are speciated and principles of the major metabolic and fermentation tests. So I suggest that you look those over and that you look this material up in your textbook and really take good notes over this unit. Listen to it a few times because this is probably one of our tougher units. All right, I look forward to visiting with you for Unit 12.